Okay, let's unpack this. Today, we are doing a deep dive into the absolute cutting edge of health and oncology.
Right.
Our mission is really to cut right through the complexity of advanced data science, AI, multi-omics,
All the big topics.
all of them, and understand how medical science is finally achieving this, well, this unparalleled predictive precision.
And we're starting with a very human, very high-stakes clinical problem. We're focusing on the most common form of breast cancer.
Estrogen receptor positive, HR2 negative early breast cancer.
Exactly. It makes up something like 70% of all cases.
And that's the challenge, right? You have this massive population of patients, and the clinician faces this agonizing, life-altering decision.
Who gets chemotherapy?
Who absolutely needs that heavy burden of chemo, knowing the severe quality of life hit that comes with it, and, who can safely skip it?
And the traditional methods, they just leave you stranded.
Stranded in a massive gray zone of uncertainty where the answer is truly unclear.
Precision is everything here. And frankly, we just haven't had enough resolution. Our sources today tackle this problem by showing two revolutionary, really game-changing approaches.
It's a dual approach. So first, we're looking at how artificial intelligence can assess relapse risk right from a standard pathology slide.
Just looking at the morphology, the structure.
The architecture of the tumor and its surroundings. And second, we dive deep into the tumor's genetic blueprint using whole genome sequencing, or WGS, to decode the molecular chaos inside.
And the ultimate goal here is moving beyond that old reductionist thinking.
To viewing the human being as a complex, evolving ecosystem. Okay, so let's start with the immediate clinical win, the AI tool, this R-risk prognostic model. This is a deep learning system that takes a digitized image of the entire tissue slice, the whole slide image, or WSI from the primary tumor.
And from that image, it predicts a patient's 5-year metastasis-free survival.
What's so powerful here from what I'm reading is that the AI isn't just confirming what a pathologist might already suspect.
No, it's functioning as a powerful, and this is the critical part, an independent prognostic factor. It's spotting patterns that are either, invisible or certainly too complex for the human eye to consistently quantify.
And when they integrated this AI score with traditional clinical factors, age, tumor size, node status, that kind of thing, the combined model saw a big jump in predictive power.
A significant jump.
Can you walk us through those metrics? The C-index jumped from 0.76 for the clinical factors alone up to 0.81 for the combined model. What exactly is a C-index, and why does that tiny-sounding 0.05 increase matter so much?
Right, it does sound small, but the C-index, or concordance index, is essentially a measure of reliability.
Okay.
Think of it as how one model can distinguish between two random patients, one who will have a relapse and one who won't. A C-index of 0.5 is a coin flip.
And 1.0 is perfect.
Perfect. So moving from 0.76 to 0.81, that represents a profound clinical improvement in the reliability and the confidence of that prediction.
So we're not just moving up a little notch, we're fundamentally reducing the ambiguity.
Exactly. It means moving potentially dozens of patients out of that terrifying, ambiguous intermediate risk group.
Where the decision to use chemotherapy is just agonizing.
It is. The AI provided such clear data, it was capable of what's called clinical dichotomization.
Yeah.
It splits patients into a genuine low-risk group list, then a 5% chance of relapse, and a high-risk group.
And the difference between those two groups was staggering, a hazard ratio of 8.01. For the listener, what does an HR of 8.01 mean in practical terms?
It means the high-risk group, identified by the AI, is 8 times more likely to metastasize within 5 years than the low-risk group.
8 times.
That level of separation is a clear mandate for totally different treatment strategies.
That is massive. But here's the conceptual shift for me. The AI isn't just looking at the cancer cells, like their size or grade.
No.
It's looking at the whole neighborhood. The sources highlight that it also picked up on features associated with decreased risk. What were those protective features?
It's focusing on the entire tissue microenvironment. The model picked up on things like the presence of vascular structures, the formation of fibrosis, which is just scar tissue, and even specific patterns in the adjacent normal tissue.
So it's integrating information about the body's protective response, not just the enemy's aggression.
Precisely. It makes the AI less of a high-tech slide counter and more of a subtle ecosystem analyst.
And the logistical advantage is undeniable. It uses the standard slides already made for every diagnosis. It fits right into the workflow.
No special biopsies needed. It's the definition of leveraging existing infrastructure.
Okay, so from that macro view of tissue, let's go to the hypermicro view of the genome. Whole genome sequencing.
Right. And we are looking at an unprecedented cohort from Kubrick 264 Korean breast cancers analyzed in incredible depth. WGS maps the entirety of the tumor's DNA.
And because the median age here was pretty young, 44 years old, it helped researchers find potentially distinct genomic drivers, and they found 4 new ones.
They did. 2 that really stand out are BCL1B, which is involved in chromatin remodeling, basically reorganizing the DNA's packaging, and RRE1, which regulates cell growth.
And finding a new driver that's the holy grail.
It's incredibly exciting because it's the first step toward developing a completely fresh, targeted therapy. It opens up whole new treatment pathways.
This genomic view also gets into the question of stability. The sources highlight the mutant allele tumor heterogeneity score, the math score.
Right.
When I hear that, I just think genetic chaos. Is that what it's telling us?
Genetic chaos is a perfect way to describe it. The math score quantifies the genetic diversity inside the tumor. If all the cells are clones, the score is low. If they're all different, the score is high.
And a high score is bad news.
Very bad news. A high math score 40 or greater correlated with significantly worse overall survival.
And it had a huge implication for targeted drugs.
Absolutely critical. A high math score predicted significantly worse progression-free survival for patients on CDK4/6 inhibitors, a hazard ratio of almost 3.0.
So the takeaway is, if the tumor is chaotic, targeted drugs won't work for long.
Exactly. There's so much variation that resistance is basically pre-programmed into it. It forces an immediate strategy change.
The sequencing also revealed some striking population genetics. They found a germline deletion inherited involving the Epoactria B genes.
And it was present in nearly a third of the Korean cohort, 31.8%.
Compared to only 8.5% in European populations. Why does that 4-fold difference matter to a patient?
It matters because carrying this deletion leads to a significantly higher tumor mutational burden, or TMB. And TMB is a really important clinical marker, often used to predict how well a tumor might respond to immunotherapy.
So a population-specific finding like this could fundamentally change treatment guidelines for that population.
It could. It's a major marker. And the chaos isn't just small mutations, it's physical structure.
They detailed how rearrangements can drive resistance.
Yes, this recurrent translocation of chromosomes 8 and 11. Segments literally swap places, bringing the CCND1 and FGFR1 genes close together.
Which upregulates progression pathways.
And, crucially, is associated with reduced benefit from standard aromatase inhibitors. A physical miswiring in the DNA directly causes resistance.
Wow. And to wrap this up, the most elegant, if terrifying, example of this evolution is the real-time resistance they saw with PRP inhibitors.
This is truly evolution at fast forward. There was a patient who responded beautifully to the PRP inhibitor talazoparib, but then resistance developed.
Ow.
The tumor acquired a tiny 8 base pair deletion right near a pre-existing BRCA1 deletion. And this new small deletion effectively restored the DNA repair pathway.
The tumor fixed itself.
It literally fixed its own vulnerability. It went from being deficient back to proficient, and the drug just stopped working because the target had repaired itself.
That micro-precision is profound. From the AI analyzing scar tissue to WGS capturing an 8 base pair repair job. But the bigger narrative here is this philosophical shift.
It is.
Moving from reductionist thinking, the tumor as an isolated problem, to systems biology, viewing the human as a complex, evolving ecosystem.
That is the pivotal shift. The editorial insight captured it perfectly. If you just focus on the smallest details, you never get the big picture right.
For decades, we just dissected disease into isolated parts. The rogue gene, the single bad cell.
And now we see it as a systemic disruption of an intricate biological web, a cascade of failures.
To properly map that web, we need multi-omics. We've established the genome is the blueprint. But what are the other crucial ohms that give us this dynamic picture?
So think of it like a house. If the genome is a static blueprint, then epigenomics are like the annotations, the sticky notes on the blueprint saying which rooms are open and which are closed.
Without changing the blueprint itself.
Exactly. Then you have the active work. Transcriptomics captures the messages, the cellular activity like the email traffic inside the cell.
And proteomics.
That's the physical machinery, the functional proteins that are actually doing the work. And metabolomics analyzes the chemical fingerprints, the fuel and waste from all that work. It's the output signature.
And finally, the external influences on the system, which is our microbiome.
Our microbial partners. They influence everything from metabolism to immune function to how we respond to cancer treatment. Integrating all these layers is the essence of systems biology.
But even that whole multi-omics map is incomplete without the environment we live in. That brings us to the missing piece, the exposome. How is that defined?
The human exposome is a huge concept. It is the compilation of all non-genetic factors that interact with us over a lifetime.
Physical, chemical, biological.
And psychosocial. Chronic stress, social isolation, air quality, water quality. It's a massive driver of gene expression, and it correlates with the missing heritability in disease, all the risk we couldn't explain with just genetics.
So the core idea is the gene-by-environment interaction, GXE.
Yes. These interactions are mandatory biologically, and they're strongest during critical windows of susceptibility. And just like we have a polygenic risk score for genes, we now have its analog for the environment, the polyexposure risk score, or PXS.
And all this together, multi-omics, wearables, AI, it finally provides the infrastructure to realize Dr. Leroy Hood's 4P medicine.
Predictive, preventive, personalized, and participatory. It's moving from theory into actual practical application.
Speaking of practical application, you can't map a dynamic ecosystem like this with static measurements. A single blood draw just gives you one snapshot.
You need high-resolution data. And that's where high-frequency microsampling comes in. We're talking about small, hourly collections combined with consumer wearables like CGMs and smartwatches.
This is the enabler that lets us see how molecules fluctuate in near real time.
It is.
And this high-res data gave them immediate, specific discoveries about baseline health. They identified 332 circadian molecules.
Correct. Molecules with clear, defined daily patterns that define what a healthy baseline looks like. And what's interesting is that these were mostly lipids tags, PCs, LPCs.
So our fat metabolism is fundamentally governed by our daily clock.
It seems to be. They also measured personalized response kinetics with an Ensure Shake study. They measured the dynamic response to nutrients after a standardized meal.
And they found differences between people.
Big differences. They found one group responded significantly more slowly to the nutrient load than the other. It's not just what you absorb, but how fast you process it. You miss that timing detail completely with a single morning blood draw.
And for clinical use right now, microsampling can track pharmacokinetics, how a drug moves through the body.
They use the example of salicylic acid clearance from aspirin. They track its concentration remotely over 24 hours in one person, showing that precise drug monitoring can now be done from home.
Okay, here's where it moves toward the holy grail suggesting causal relationships. They used a lag core algorithm to find the specific delay between events. What did that reveal about, say, glucose and hormones?
The lagged core algorithm looks for a correlation that gets stronger when you shift one data stream in time relative to another. So A happens and then 10 minutes later B happens.
I see.
And they found that CGM glucose precedes the concentration of C-peptide, a marker for insulin secretion, by exactly 10 minutes.
Which is strong evidence that rising glucose causes the insulin secretion.
Extremely strong evidence, not just a correlation. And the link between glucose and inflammation was also startling. CGM glucose strongly correlated with several major pro-inflammatory cytokines.
The immune system's signaling molecules.
Right. It indicates that glucose, which we think of as just fuel, can cause a rapid, specific pro-inflammatory response. Your blood sugar is tightly tied to your immune status.
And it also confirms that the simple data from our wrist is actually meaningful.
Absolutely. Step count and heart rate from wearables were highly associated with changes in the deep multi-omics data. It proves those simple measurements truly reflect profound physiological status.
Okay, so moving from the bedside to the database, AI's role as a master integrator goes way beyond the clinic. It's being used to accelerate knowledge generation by analyzing patents.
It's a massive different kind of resource. PubChem expanded its co-occurrence analysis, which usually looks at biomedical literature, to the Google patent research data.
And this includes the full text titles, abstracts, claims, descriptions. It's a huge expansion of coverage.
It is, but patents are legal documents. They are written vaguely, often listing dozens of common chemical solvents prior art that aren't the actual invention. It creates overwhelming noise.
The initial analysis was just clogged with water and ethanol.
Exactly.
Yeah.
So they needed specialized algorithms to filter it. They introduced something called the informativeness weight.
Okay.
It decreases the contribution to annotations from patent sections that list a huge number of similar things. If you list 100 solvents, that section gets weighted down as less informative.
And then they added the ultimate noise cancellation tool, the exclusion list.
A curated list of 262 common compounds, water, ethanol, sodium chloride, general stuff that's everywhere, and just overwhelmed the results.
And the power of this filtering is really the key takeaway here. What did the exclusion list do when they queried for neoplasms for cancer?
It was a conceptual victory for the algorithm. Before the exclusion list, the top chemical neighbors for neoplasms were generic things like water and ethanol, totally useless. After applying the list, the top neighbors shifted dramatically to specific high-relevance anti-cancer drugs. Doxorubicin, paclitaxel, cisplatin. The AI converted chaos into targeted relevance.
So this confirms that patent data, once it's properly filtered, becomes incredibly selective for genes and diseases, focusing intensely on commercial targets. It's a valuable counterpoint to purely scientific abstracts.
So we've covered these two converging pathways of precision health. On one hand, you have micro-precision in diagnosis, with AI and WGS refining clinical risk in cancer with really unprecedented clarity.
And on the other, you have this macro-precision in monitoring with multi-omics, exposomics, and microsampling capturing the dynamic ecosystem of health.
Both are miraculous breakthroughs.
Yet both rely absolutely on intense standardization to move from a research paper to global reliability. We noted that even the AI tool, which just uses a standard slide, requires a 30-slide calibration protocol for every single new lab before it can be trusted.
And that logistical challenge is mirrored on the systemic level. I mean, interpreting chaotic WGS data, integrating the messy inputs of exophthalmic data, the sheer labor involved in ensuring quality is immense.
And this brings us to our final thought for you to mull over. We are relying entirely on these complex, high-tech systems to deliver these individualized insights. As we try to scale this up globally, how do we ensure that the immense necessary human effort required for data quality, from the technician carefully calibrating that AI tool to the data scientist integrating chaotic genomic and environmental data? How do we ensure that doesn't remain the single greatest point of failure in making personalized health truly universal?